For DNA replication to begin, the replication machinery must determine where replication starts, where is its origin. And humans have a number of proteins which function in binding to DNA to determine the origin of replication. Since DNA is so highly coiled, molecules known as topoisomerases must twist and untwist the DNA to prevent supercoiling. Some make single cuts in the DNA strands, some make cuts in both strands. DNA topoisomerases seem so different from each other when comparing one group to another. It suggests that much of the replication machinery had not yet been perfected in the last common ancestor of all life on Earth, and so that each group has then specifically developed new molecules in order to do this. Some are specific to viruses, suggesting that some of them even originated in viruses. Humans have a variety of these proteins. Mutations in some of them affect our sensitivity to drugs. A number of proteins in the human genome bind to single-strand DNA to stabilize it during replication. Humans have 3 genes coding for primases, the enzymes which will start a short RNA molecule to which DNA polymerase will add DNA nucleotides in DNA replication. So DNA replication begins with short RNA molecules known as primers, and we have 3 enzymes called primases which form these. DNA polymerases add new DNA nucleotides to growing DNA strands. Humans have a variety of these enzymes. Bacteria may have 3. And mutations in some of these enzymes can leave people more vulnerable to cancer, less likely to be able to repair their DNA, especially after ultraviolet light damage, and some can even cause premature aging syndromes. Many other proteins of the human genome function in DNA replication, such as the ligase, which unites ends of fragments to make continuous chains, and then some proteins which are needed for the termination of replication. The ABC cassette transporters are related to the helicase superfamily of genes, and it is thought that these 2 superfamilies have descended from the same ancestral genes. Helicases typically use ATP to unwind DNA or RNA. The ATP binding regions of both ABC cassette transporters and helicases share homologous regions. This gene family also includes the REC-Q-like family, which unwinds DNA, the dead box, DEAD box, RNA helicases, which can unwind RNA regions and act as splicing factors, eukaryotic translation initiation factors, ATRX, which is a member of the helicase superfamily, which can result in mental retardation and even sex reversal. Okay, so ATRX is involved in gender formation and in placental mammals, and it is required for the effect of SRY. And the REC-A family, this REC-A protein is essential for the recombination which occurs between sister chromatids in gamete formation, and which is also used in the repair of some mutations.